Chinese science turns waste into treasure, using carbon dioxide to produce artificial starch. The challenge of feeding the growing global population while minimizing environmental impacts has prompted scientists to explore innovative solutions. A research achievement in China has shocked the world. It turns out that domestic scientists have successfully turned waste into treasure, using carbon dioxide to produce artificially synthesized starch. A large part of the crops planted by humans are used to obtain starch, such as corn, wheat, rice, etc. The main nutrient is starch. Plants can use photosynthesis to produce starch, while animals rely on consuming plants for energy. If not consuming enough starch, it may even affect human health. At present, starch is mainly synthesized by green plants through photosynthesis to fix carbon dioxide. In the photosynthesis of plants, carbon concentration is necessary to generate more sugars. The increase in carbon dioxide concentration allows plants to add it to the 3-carbon molecule phosphoenolpyruvate, PEP, to participate in the reaction, thereby fixing carbon dioxide in mesophyll cells. This reaction is called the enzymatic catalysis of PEP carboxylase, which produces a 4-carbon organic acid oxaloacetic acid. Carbon concentration enhances the photosynthetic capacity of plants under catalytic action and allows crops such as corn, sorghum, and sugarcane to generate more sugar under high light and high temperature conditions. Almost 90% of plants use 3-carbon molecules for carbon concentration fixation. The photosynthesis of plants is a series of very complex chemical processes, with over 60 involved steps. In terms of the efficiency of the entire photosynthetic process, the photosynthetic efficiency of plants is usually between 3% and 6%, and unconverted light energy will be dissipated in the form of heat. And such conversion efficiency will vary with changes in temperature, light intensity, and carbon dioxide concentration. So, besides photosynthesis, is there a more efficient way to produce starch from carbon dioxide? To solve this problem, researchers from the Tianjin Institute of Industrial Biology, Chinese Academy of Sciences have designed a new pathway for non-natural carbon dioxide fixation and artificial synthesis of starch with 11 main reactions from scratch. For the first time, complete synthesis from carbon dioxide to starch molecules has been achieved in the laboratory. The research team adopted a building block approach, in collaboration with the Dalian Institute of Chemical Physics, Chinese Academy of Sciences, to use chemical catalysts to reduce high-concentration carbon dioxide to carbon-1, C1, compound under high-density hydrogen energy. Then, by designing and constructing a new enzyme for carbon-1 polymerization, the carbon-1 compound was polymerized into carbon-3, C3, compound based on the principle of chemical polysaccharide reaction, and finally optimized through biological pathways, polymerization of carbon-3 compounds into carbon-6, C6, compounds, followed by further synthesis of linear and branched starch, CN compounds. Inspired by natural photosynthesis, the overall design concept is to use high-concentration carbon dioxide generated by energy factories as raw materials, which are then converted into energy using solar cells, and undergo hydrocarbon synthesis to ultimately generate starch. This sounds simple, but in reality, scientists face a lot of problems, the first of which is how to make an effective practical transformation. Artificial synthesis is not like nature. Natural synthesis in plants does not require additional variable factors to participate, and the entire photosynthesis process is a perfect closed loop. In addition, the participation of each biological enzyme is very coordinated, and the enzyme-catalyzed reaction will not have any problems, but artificial situations cannot guarantee 100% error-free. Therefore, scientists first made a basic summary of the adaptation of biological enzymes. Due to the imperfect nature of artificially selected biological enzymes, some catalytic reactions may produce contradictions. Therefore, selecting the appropriate enzyme pairing is also crucial. The research team divided carbon coordination into single carbon compounds, 3-carbon compounds, 6-carbon compounds, and multi-carbon compounds. Find the most suitable combination for each type of carbon complex. Firstly, there is the part of light energy synthesis. As it is artificially synthesized, scientists have used solar cells to simulate it as a plant and absorb light energy for charging. Then the electrical energy is sent to a device that can perform electrolysis, with water as the electrolysis component. After electrolysis, water generates oxygen and hydrogen gas, which are then chemically reacted with carbon dioxide and hydrogen gas. Zinc oxide is used as a catalyst to obtain methanol and water. Methanol is a key synthetic substance in this process, just like NADPH and ATP mentioned earlier, which are the main energy substances required by organisms. The second stage enters the Calvin cycle process, where scientists use computer simulation analysis to arrange and combine the prepared biological enzymes and carbon compounds, and find the key ratio among over 6,000 combinations. With suitable biological enzymes, carbon coordination, and the energy required for synthesis, the next step is to place them in a bioreactor for higher-order reactions. In this process, the synthesis of energy reacts with methanol to generate dihydroxyacetone, DHA, a 3-carbon compound that undergoes the participation of biological enzymes and ultimately generates starch under the Calvin cycle. Compared with natural starch synthesis, the efficiency of artificially synthesized starch is very high, taking only 4 hours to synthesize starch. If we take corn, a high-starch economic crop, as an example, the growth cycle of corn is about 4 months. Looking at it this way, we can make a clear judgment. The starch synthesis rate of this artificial pathway is 8.5 times higher than that of corn starch synthesis, taking a big step towards designing natural and surpassing natural goals, providing a new scientific basis for creating new functional biological systems. Under sufficient energy supply conditions, according to current technical parameters, the theoretical annual starch production of a 1 cubic meter bioreactor is equivalent to the average annual yield of corn cultivation on 5 acres of land in China. This achievement has made it possible to transform the traditional agricultural planting mode of starch production into an industrial workshop production mode, and opened up new technological routes for the synthesis of complex molecules from carbon dioxide raw materials. This achievement has opened a window for industrial workshop manufacturing from carbon dioxide to starch production. If the process cost of the system can be reduced to be economically feasible compared to agricultural cultivation in the future, it will save more than 90% of arable land and freshwater resources, avoid the negative impact of pesticides, fertilizers, etc. on the environment, improve human food security level, promote carbon-neutral bioeconomic development, and promote the formation of a sustainable bio-based society. Despite the success of this technology, we cannot ignore the fact that although the synthesis efficiency of artificial starch is higher, it requires a large amount of carbon dioxide and electricity, while plants do not require so much energy to supplement and can also release oxygen. In addition, the artificial synthesis of starch is carried out in the laboratory, which falls far short of the production standards in terms of scale. Therefore, from an economic perspective, this technology still needs further improvement. Our research team also stated that in addition to economic costs, the current artificial starch synthesis technology also needs to improve the efficiency of electricity utilization and carbon fixation conversion of starch, and this efficiency must be increased dozens of times in order to have the opportunity to compete with modern agriculture. So this technology still cannot meet the social needs of the Nobel Prize, so we still have a long way to go.